Well, thank you everybody for coming. I'm very humbled to hear that so many people are signing up today. I agreed to do this for the first go around, figuring that I'd be the guinea pig, and if it didn't work, well, it was the first time. But I hope it does work out well, because it is a story that I am excited to share with everybody, especially the folks who are affiliated with Washington's National Park Fund, because this project really would not be what it is today without your support. Right in the initial phases of the project, we lost a significant chunk of funding, and the park fund stepped up and filled that gap and enabled us to do the work that we did, that I will share with you now. So should we get going? Okay, so I do not have the ability to use Zoom on my Park Service computer, so we're going to be kind of working remotely. I sent this presentation over to Park Fund, and I'm going to ask them to kind of go through the screen, so it might be a little glitchy. But I'm going to be talking about the project to restore fishers to Olympic National Park. And then there also is ongoing programs at Mount Rainier and North Cascades, but this is a project that happened first. Okay, next slide, please. So just as Lori said, fishers are a member of the weasel family. They are kind of the fifth. It's a pretty diverse family, ranging from the smallest is the little least weasel to the largest is going to be the sea otter. They look kind of more closely related to marten, which you might be more familiar with. They're a forest carnivore, but actually they are most closely related to wolverine. So they're kind of like a smaller wolverine that likes to live in trees. They do live in their habitat requirements are low to mid elevation forests. And one of their key characteristics is as they move around, because they're kind of mid-sized carnivores, they need some places to kind of rest and get away from larger things that eat them. So they use tree structures like limbs or platforms to rest in. Next slide, please. For their size, they have really large home ranges. A female fisher in our area has a 25 square kilometer home range, about 3 square miles. And this graphic actually shows the home ranges of some fishers in California, and the smaller polygons are female home ranges. You can see that they're kind of adjacent to each other, but they don't really overlap much. And then the male's home ranges are about 3 times that size, and typically a male home range will overlap several females. Next slide, please. One of their key habitat requirements is that they have their young in tree cavities. So the forested stands that they live in, the trees have to be old enough for the decay processes to occur and form cavities that are big enough for a female fisher to slip in and out of and park up to 4 kits at a time. And that's often a limiting factor on the landscape. You know, they don't live very long. Usually about 10 years is kind of the record in the wild. They don't give birth very frequently, so they are vulnerable, their life history. They don't reproduce very quickly. Next slide, please. They once occurred throughout the forest, boreal and temperate forest in North America. They are a North American species only. And so on this map, you can see the orange shows where they used to occur. And then their range was greatly restricted around the turn of the century to just those areas remaining in blue. And the loss of the population was primarily driven by overharvest for fur. Their fur is analogous to the Russian sable. And at the time that massive trapping was occurring, there were no regulations. And by the time regulations came in, it was often too late to save the population. And that was the case in Washington State. So before we started this project, we felt pretty securely that there were no fishers left in Washington State. There was a remnant population down in Southern Oregon and Northern California and in the Sierras, but there weren't even any populations in British Columbia that abutted Washington. Next slide, please. So we, working with our state partners, started really evaluating the status of the fisher in '88, and there's a long journey in planning and compliance that culminated with the EA that I worked on with my partner, Washington Department of Fish and Wildlife, that got finalized in 2007, I think. And in that analysis, they evaluated, is there still suitable habitat for fishers in Washington state? And the answer was yes. And then where are the best places to reintroduce them? And the best place for reintroduction in that analysis was the Olympic Peninsula, Primary and Olympic National Park, because we had the large chunk of habitat. And the plan was that if we were successful, then they would continue on the project in the Cascades. Next slide, please. So once we completed our environmental compliance and had our partners all lined up, we started actually putting it on the ground in 2008. And this is just a slide that shows all the steps that were all put together. So what we did in your upper left-hand corner, I think that's your left hand. Anyway, we contracted with people who were still fur trapping for fishers up in British Columbia, and we asked them to divert their harvest from the fur harvest to live trap for us. And we had a lot of people sign on and do it. So we had these trappers go out, and when they caught a live fisher, they called a trapper coordinator who took them back to their place in Williams Lake. And when they had about 10 fishers, we would go up and work with the veterinarian to process them. And that's me kind of in the slide with the BC wildlife vet. They all got checked. And one of the key things that they had to have is they had to have enough teeth so that they could forage efficiently when we released them. And everyone that passed the check was put in a box in the back of a truck. We drove them down overnight and released them the next day. Next slide, please. So over the 3 years of the translocation, we ended up releasing 90 fishers. In 2008, we got a little bit of a late start. We only released 18, but we got more organized with the trappers, and we really ramped it up in 2009 and 2010. So this graphic, it's pretty small on my phone. I hope you guys can see it, but up in British Columbia, the different colors show which animals were caught in the different years. And then down on the Olympic Peninsula, you can see where the different release sites were over the years. Next slide, please. So all the fishers that we released had a radio transmitter collar on them, so we could follow them to see, is this going to work? The first question was, are they living? And then if they survived, were they staying in the release area, and were they able to establish home ranges? And then we also wanted to know, if they are living and hanging around, are they able to effectively reproduce? Because you need all those 4 things for a population to establish and say that you have recovery. Because we are a large wilderness park, we figured there's no way we're going to follow these animals on the ground. So early on, we committed to doing aerial telemetry to find the animals. And everybody knows our weather. It was a little challenging, but we're fortunate to have a really good pilot in Port Angeles that we were able to fly, well, just about every other week, and sometimes a couple times a week, depending upon what was going on with the weather and if it was a denning season, we wanted to fly more often. Next slide, please. So early on, we were first started tracking, it was like, oh, thank goodness we have a budget for doing aerial telemetry, because they started moving really quickly. So this graphic and the next one are all going to be about the same. Each individual color shows a different animal, and I just kind of connected the dots of where they moved to. And the females have the squares, and the males are the red circles. So you just kind of can track and see where they went. And we thought that fishers would be restricted in their movement by rivers or the snowy ridges, and that did not hold them back. Some of them started traveling really far, really quickly. Next slide, please. When it was all said and done at the end of the year, all the animals kind of did something different. So there was no rhyme or reason to where they went. So this graphic shows the movement over a year of the first animal we released, which is female 6. Remember her name. We released her in the Elwha at near the Elwha campground, I think, at the ranger station. That's right. And she hung around that area for a little while, and then picked up stakes and moved over to the upper Dosey. She actually hung out there for a year. In the second year, which is not shown here, she decided that wasn't the place to be and she moved back to the Elba. Next slide, please. Another female, female 4, remember her name, she gave me a heart attack because soon after we released her, she moved to outside the Port Angeles airport for a while, which I thought that's not good. And then she picked up stakes and, you know, kind of came to her senses and moved deep into the park in the upper Queets. And I went, phew, she's going to be safe. And then she decided to move. And she moved actually to right outside Port Angeles. And if you know where 101 takes that bend before you get into town, when you're near Walmart, that's where she lived for 3 years. Next slide, please. Other females just bounced around the entire park, going on Forest Service land for the entire summer. And some of them didn't settle down until October. And so this is the female 17 who just bounced all the way around and finally ended up in the lower Queets. Next slide, please. And the males were even harder to figure out. This is the movements of male 11. Remember him? We'll get back to him later. He stayed around the area for a little while, and then we lost him and couldn't find him for over a month until we finally picked him out at Nia Bay. So he totally left the study area and wasn't even in the coastal strip. I think that the only thing that stopped him from going back to Canada was the saltwater. Next slide, please. So at the end of the first year of monitoring, we had pretty good survival. This graphic shows the home range establishments of all the fishers that lived for a year. The females are outlined in kind of the greenish teal boxes, and the males are shown in the red circles. So, you know, what's the problem here? Well, the girls are in the mountains and the boys are in the beach, and that really doesn't lead to successful reproduction because we need to have, you know, that to happen for the population to reestablish. The only one male that we thought might be hanging around where the females settled down is male 9, who's kind of shown in an open circle around Hurricane Ridge. His radio transmitter went dead, so we didn't know if he was still alive. We hoped he was because that's where the females were, but we did not know until later. Wait, next slide, please. So in 2009 and 2010, as I said, we continued to release fishers. We kind of hoped that the second and third cohorts would hang around. Once there was fishers on the landscape, they maybe would not wander around so much, but that really didn't happen. Each year that we released animals, they continued to just, some stayed put and some wandered around. And we did do an analysis to try to figure out what kind of factors predicted how far animals moved, and there really was nothing that was evident. I think it was just individual choice. Next slide, please. But we did find that in 2009, those males, male 11 and another male 14 who went to Ocean Shores, actually returned to the release area where the females were during the breeding season in 2009, but then went back to where they were before. So I guess they just like being on the beach. Next slide, please. So at the end of the first phase of the Fisher Project, this is kind of what we ended up with. And after 2010 and into 2011, there were some males that kind of settled down near where the females were, but not a lot. And for a small animal that's so widely dispersed across the landscape, we had real concerns about the implications of this on the population, you know, successfully breeding and reestablishing itself. Next slide, please. We did do as best we could to go in on the ground using radio telemetry and try to locate den sites of females. This graphic shows all the females that we were following who did restrict their movements during that denning period in May and June. But we were only able to document the den sites of those females that are shown here with the dots on the squares. The first fisher that was documented breeding in Washington State in over 50 years was female 7. And this is a screenshot of her moving her kits at her den site. And she denned really near Hurricane Ridge in the Morse Creek drainage. Next slide, please. So over the 4 years of radio telemetry, we only documented denning or confirmed denning in 7 females. And that's what's shown here on the graphic. So we found 3 dens in 2009. The first one was at female 7, as I mentioned, who did have 4 kits, which is the largest litter size of fishers on the West Coast. So that's encouraging. If they could find a mate and breed, they would be successful. The other 2 females that denned that year, female 22 and female 33, they actually came down pregnant from British Columbia, and then were released in January and then denned in the following spring. Same situation happened in 2010. That female 4 that settled down outside Port Angela, she finally did den in 2010. So she was in the landscape for over 2 years before she denned. And then we were able to, we didn't have as many fishers on the air in 2011, but we did document that 1 den then. So you see those females whose numbers are highlighted in pink. One of the concerning things to us too was all those 3 females did die during the denning period. They were all predated. So although we had 7 successful dens, 3 ended up not making it because of female mortality. Now, the one with the asterisk on it, female 88, we actually got on that den site quickly and were able to rescue her 2 kits. They were sent to Northwest Trek to raise kind of away off display to maintain their wild characteristics, and we actually successfully released them in October of 2010. Next slide, please. So of the fishers that we released, the 90, we had fairly decent survival the first year. It was about 80% survival of the females. 2009 was not such a great year. So overall, about 50% of the females survived. We had a much higher survival rate of the males. And the major cause of mortality was predation. It's going to be bobcats and cougar were the main predators of these females, but also some roadkill and some drowned. Next slide, please. So at the end of the telemetry study, we had some cause for being, encouraged that the restoration is going to work, because we knew that the fishers were persisting on the landscape. We had some reproduction, but we didn't have a lot that we could document. And so we really couldn't say whether the restoration program was successful or not. Next slide, please. So we had planned all along to move into the next phase of the research, was to follow up the monitoring with non-invasive methods using cameras and hair snares to see, are there still fishers on the landscape? If so, where they are. And so the cameras, we would just bait them in and take a picture and say, yes, it's a fisher or not. But those hair snares are really important because we wanted to grab DNA. And if we could grab DNA and if it would amplify, send it to the lab and they could tell us who it was. So while we still had animals on the landscape and we knew where they were, we tested out all these different camera configurations and bait configurations in their home range to see what would draw them in the best. And the best that worked was these little hair snare cubbies and chicken. So that's what we went with. And we did that project from 2013 to 2016. Next slide, please. And if you remember that map where I showed you where all the fishers ended up dispersing at the end of the telemetry study, we knew from the get-go that we could not do any meaningful measurement of what's going to be the Olympic fisher population if we stuck in the park. So our study actually covered the entire Olympic Peninsula. So those little hexagons that you see on your screen right now are all the areas that we separated out for sample. Now, we do have a gap in the Quinault Reservation. We did not have permission to work there, and we didn't sample on the top of Mount Olympus, but we did go everywhere else. And we focused our research on the Olympic National Forest north to the Straits of Juan de Fuca, but we had some expansion if we could bring in partners out into the Quipper Peninsula near Port Townsend, and then down south to Highway 12 to Olympia and Aberdeen. Next slide, please. So in each of those hexagons, we put 3 sampling stations, and this graphic shows what was at each sampling station. And this is one of our text kind of recording data as she set it up. So there was a camera on a tree that was focused on chicken that was nailed to an opposite tree. And above that, there was a sponge that was, or actually we used moss that was soaked in skunk lore to bring them into the site. And then the fisher would come to try to get at that chicken and get its picture taken. And the chicken was wrapped in chicken wire, so they couldn't just grab and go. And then at the base of the tree was this hair snare that was also in the frame of the picture, so we could see which animal was coming in and out of the picture. We set up these camera stations throughout all those hexagons that I showed you in the study plan. So it was a pretty massive project. When a camera was set up, we checked it on 3 2-week intervals, so they were out for 6 weeks sampling. And we had known from a prior sample that if a fisher was in the area, we had a greater than 95% chance of capturing on the camera. So if they were there, we were going to get them. Next slide, please. And as I said, this is a really big endeavor. We could not do it all by ourselves. So we had a lot of help from just about every tribe in the region. Took on portions of the study that they were going to do. Also, Washington Department of Fish and Wildlife did part of it. We had an NGO out Port Townsend that did the Peninsula. So that was how we were able to sample the entire Olympic Peninsula. The park staff did the national forest in the park, maybe about 2/3 of the sampling, but the rest was done by partners. Next slide, please. So this is the results. In 2013, our first year, we picked up fishers in 9 of the 52 hexes. We got a slow start that year. I don't remember, that was one of the government shutdowns and the sequester, so it slowed us down a little bit. So this slide and the rest of the slides are all going to look the same. The white hexes are where we sampled and didn't get a fisher. The black hexes are where we sampled and got a fisher that year. And the red numbers are the individual identification of those animals. If it has a number, that means we figured out who it was, and based on the hair analysis. And if the number starts with an M or an F, that was one of the ones that we released. If it starts with an OPF, that was their offspring. So one of the animals that we got, if you see on the Northern Olympic Peninsula, was F06. So that was the first animal that we released in 2008, and she was still alive in 2013 in the same area, same home range, which was really exciting for us. And what was also even more exciting, we followed her for 3 years and she never denned that we could document. Well, we picked up her son all the way down near staircase. You can see that 005 animal, that's actually her son. So that was exciting to be able to do that with the DNA analysis. Next slide, please. 2014 was a bit of a grim year. We only got fishers in 5 hexes, all kind of down in the southern portion of the range. But we like kind of the little rainbow in the overall cloud was that one of the animals that we picked up was M101, which was actually one of those orphan kits that we released. So he was still living 4 years later. So we knew that it was a successful endeavor. Next slide, please. In 2015, the number of fishers that we were able to catch picked up. And what's interesting to us is that we got our first second generation fisher. So the lower photo is of the female that is at Claylock, who was the offspring of one of our release fishers. And then her daughter is the one that's shown up at Lake Ozette. So we were able to document that they're not only did our founders were they successfully able to breed, but that their offspring is also surviving. Next slide, please. And then this is the last year that we did in 2016, again, continuing to pick up offspring and then their progeny. Also for the first time picked up fishers moving out of the primary study area down in the managed land in the southern peninsula. And then if you look at that picture really closely that I showed on the graphic, that's from the hex up in the Solduck, and there's actually 2 fishers in there. We have a male and a female. One of them is one of the ones that we released in 2009, and another is a granddaughter of female 7 that we released in 2008. Next slide, please. So this is the picture putting it all together. So in the main occupancy study, we were able to get fissures showing up in 24 hexes, and through the DNA analysis, we were able to identify 22 unique individuals. So that, that's encouraging. They're still there. Occupancy rate was still kind of generally low. It averaged around 14%. In an established fissure population, you will have usually at least 50% or more, but they were still there in the landscape and they were still reproducing. Next slide, please. But that was not the only way that we were getting information on fissures. While we were really focused on it, we were kind of fissure central for Washington State. We asked anyone whoever sees or finds a fissure, let us know, and we'll put a camera out and try to get DNA. We got a lot of incidental observations, either roadkills or tribes are still allowed to live trap. So in the upper left-hand corner, you'll see a guy holding a fissure. That's that male 11 who went out to Nia Bay. He was actually live trapped and released. I think that was in 2015, and at that time he was 9 years old. So that shows that, okay, doing pretty well. The male in the upper right-hand corner, he was a wild one. He was actually photographed by somebody's cell phone who was taking a walk out on Dungeness Spit. A coyote had treated him. So we were just there's a whole bunch of variety of reports of other fissures that weren't in the main study that we added to our data set. Next slide, please. In addition, we had a study working with the Forest Service looking for marten from 2013 through 2016. We did not pick up any marten, but we did pick up a heck of a lot of fissures, especially on the coast. And you can see the little blue dots are all the marten stations on the coast that picked up fissures. So when you put all the different avenues of investigation together, we ended up identifying 35 fissures in the 4-year period of the study through genetic analysis. Next slide, please. So for me, who has kind of like known these animals intimately, this genetic analysis has really been a heck of a lot of fun because, I mean, some of these animals like familiar with when we went up and got them in British Columbia. And so I was able, working with genetics lab, to kind of put family trees together. So this slide shows the progeny of that female 7, that first one that we documented denning near Hurricane Ridge in 2009. She is shown here in the pink, and her daughter showed up down near Staircase, and that's a picture of her daughter. And we know that the father of that female was that male number 9. So he did live, and thank goodness. And then that daughter who set up shop down at Staircase proceeded to have several offspring also that we've detected. One that we detected up near Solduck, and the other one that just settled down right next to her mom. The other animals that are shown are the different males combination. And then we also in the female 7 had another litter with a different male that we picked up near the Lake Angeles Trail. Next slide, please. And the other family tree is from female 48, who established a home range on the Quinault Reservation. She is the mother of the female that moved up and established a home range near Claylock. And then that female, in turn, has had 3 different fissures that we have picked up in this analysis. One through a mating with male 82, and there's a male and a female up at Lake Ozette, and then another male from a different pairing that's down near the Queets Corridor. Next slide, please. We're going to skip over this. We're going to do a little video in a bit. So, being able to do this genetic analysis was really exciting in a way because I could figure out what happened to fissures, and some whose fate we didn't know. They showed up in the genetic analysis, so we knew they had lived longer than we had thought. But the flip side was, in all those stories, I was looking at those fissures, it was the same 4 females that were showing up in over half of the progeny who we could identify their parents. And that concerned me because although we released 90 fissures, it doesn't look like all 90 of them were able to successfully breed. And what we want is to have a bunch of individuals breeding and passing on their genetic makeup into this establishing population so that we can maintain genetic diversity which is important for resiliency in the population. So the graphic over on the right is female 4, and you can see her, this is her den site with her young in June, and she has 4 kits right there. We need a lot of animals to do that, but unfortunately, she was one of the few. We know that she had 3 different litters, but a lot of the offspring out there are from female 4 and male number 9. So I asked the genetic lab in Missoula to kind of model, okay, I'm looking at this just, you know, on my spreadsheet. Is this really a concern? Have we lost genetic diversity or not? And they came back with an analysis that said, yes, we've already lost 15% of the genetic diversity of the founders of the animals that we released. And if we don't do something, in 100 years, we will have lost half the genetic diversity in the fisher population on the Olympic Peninsula, which is concerning. So I also asked them to recommend some management interventions. You know, how many, do we need to bring in more fishers? Would that help? And if so, how many? And so this graphic with the jagged lines is showing their recommendation, which is every 10 years, just bring back about 8 fishers. And this is the model for reintroducing 8 females and 8 males every 10 years on the Olympic Peninsula. And if we do that, then we will be able to maintain about 2/3 of the genetic diversity or more through their modeling analysis. So there is a way out of this. It is concerning, but it's not something that we can't do. Next slide, please. So one of the things that I am working on with partners with the Department of Transportation and kind of throughout the region is looking at areas of connectivity of fisher habitat between the Olympic Peninsula and then the recovering population in the Cascades. And this graphic does show the green tones are fisher habitat, with the darker green, the better quality. There are patches of fisher habitat that are close to each other in the Cascades, and this analysis will help us to identify those travel corridors where we need to kind of facilitate that interchange. But that's not going to happen very soon because there are no bypass or underpasses under I-5 right now, and also the population of the Cascades hasn't really recovered. So what we have asked for in the funding call for this year is some funding to just bring 10 fishers down from Alberta and release them on the Olympic Peninsula to just bump up that genetic diversity. And then with the plan that 10 years from now, they'll either link up on their own or the populations of the Cascades will be recovered enough that we can just like trap 4 and swap is the plan. And I think next slide is my acknowledgements. So I just want to reiterate that this, although I'm talking about it today, like I don't put my name on the title for slides because this is not my project. There was a whole host of people that made this happen, and you know, we could not accomplish it without them. And I just want to acknowledge everybody, the tribes, the NGOs, and also the funding from the different agencies. Key of one is the Park Fund. The Park Fund actually enabled me to hire field crew to find those den sites early on in 2009, 10, and 11. And without that, we would have never even known where our den sites were. And that's all I have, except if we want to show the video. All right. Or are we going to go to questions?
I can throw out some questions until Sharon's able to pop that up. And here it comes. I'll ask them. I love this.
So this is a video of that granddaughter of female 48 up at Lake Ozette. And, you know, she just kept on coming back to the camera so much that we took a video up there. So, and I like this one so you can see kind of how she moves across the landscape and also how they kind of come to the bait station and, you know, take their time, which is great. So we get good pictures. And in a little while, she'll be moving into the hare snare too.
That is so cool, Patty. Patty, about how many miles in a summer do they travel? You know, we saw the lines going back and forth, and do you have any idea on that?
Well, once they settle down in a home range, I mean, they're on the move all the time. But really, the interesting thing is dispersal distance. I mean, one of our animals actually dispersed over 100 kilometers. So that was, you know, when I tell my other fisher partners about that, they're kind of surprised. We do know that one animal that released in the Quinault went all the way down to Chehalis and then came back and settled down in the Capitol Forest. But as far as how many miles they travel a day, I don't know, because they circle around quite so much while they're foraging.
Yeah, makes sense. And Patty, what role do they play in the overall ecosystem? Why is this important?
So they are a mid-level forest carnivore, and so their primary prey or species, you know, throughout their range, they focus a lot on snowshoe hare. What we found here, which was novel, in fact, I just submitted a paper on it, is that they also prey on mountain beaver. And you know, there were no mountain beaver where they came from in Canada, but they figured them out right away. So, you know, they balance or adjust what's happening in kind of the small and meso mammal community. In other portions of their range, they are the only effective or the major effective predator on porcupine. And in areas where they were trapped out, the first reintroductions were actually due people bringing fish back to control porcupine. We don't have porcupine here. But, you know, mountain beaver are an issue for some reforesting stands, and we're actually finding our fishers are not settling down in the center of the park like we thought. They're more on the wilderness edge, especially in the coastal area. We strongly suspect that's because of prey availability. They're going out into the managed landscapes and foraging on mountain beaver and then hopping back into the forest for denning.
We have just a few more minutes. Someone wants to know if they made a donation to underwrite the cost of a fisher, could they name it? And we actually have toyed with that idea up at North Cascades, and then the fires came to British Columbia and put that to a halt. But I think it could be done. And do you have any comments to that, Patty?
Well, you know what? This is where I don't like that. And I'll tell you why. I don't like to anthropomorphize wildlife. And the other thing is our experience is that working with the folks in British Columbia, they named them all. And everyone they named after one of us died. So it's like a jinx.
Oh, that's not good. That's not good.
Pattie lived the longest. She lived 9 months, but like Jeff and they don't know. So if you want to, you can go ahead. Just don't tell me about it.
We're good. We're good. Thank you.
The scientist in me is like not comfortable with that, but I understand that other people do it. It's just my personal thing. So you guys can go ahead and name them. Just don't tell me.
Okay. As we wrap up, I just want to share with everyone, first, we're getting a lot of great comments and kudos. So Patty, huge thanks for you. One of the things that touches me every time you say it is when you say, well, that's M6 or F6's daughter or granddaughter. And it's like, it just touches your heart. It really does. So thank you for that. As we begin to wrap up, I want to share with all of you, this has gone over better than we anticipated. We're very happy that so many people were so interested. Give us your feedback. Let us know what you think. Send us an email. My email is Laurie, Laurie, at WNPF.org, or Sharon, SHARON, at WNPF.org. Next Wednesday, we may be featuring a program geared more towards younger learners, so keep that in mind. We have a new webpage set up on our website that is announcing the programs as we go. 2 weeks from today, you'll want to mark your calendars. We're going to hear from Tracy Swartout and spend a day hear about a day in the life of the person who is second in command at Mount Rainier National Park. She is articulate and fun to listen to and hear her story. So 2 weeks from today, go to the website to learn more about watch for the announcements as we send them out in e-news. If you're interested in supporting the Fisher Project, give us a call, let us know. There's definitely a need, as Patty has said, we continue to support the program. If
anybody has any questions, if I didn't get to answer all the questions, you could just email me at work. I'm working at home now, and there's not much I can do, so I can answer emails.
And Patty, your email address?
It's my name. So PATTI_HAPPE@nps.gov.
Great. And then finally, as you all depart, don't forget to pick up your backpacks. Don't litter, so pick up your lunch wrappings. Enjoy the bus ride home, and thank you so much for joining all of us on our first ever virtual field trip to Olympic National Park. Take care, everyone.
Thank you.